Okay, so I have 4 o'clock. Let's do some Biology 107. We are going to get right back into the macromolecules. And last day, we covered lipids, we covered carbohydrates, we covered proteins. There was one protein slide that I hadn't finished talking about. And today we're going to talk about nucleic acids. So let's take a look at what we missed regarding proteins. Mostly I wanted to talk about this slide here, which is dealing with a disease called sickle cell anemia. And we're going to come back way at the back end of the course. We're going to talk about sickle cell anemia again, but I want to show you what it is. And it's a disease of a protein. So it's a genetically inherited disease. And if you take a look, it actually affects protein structure. So if you look at what we're seeing here is that most people have normal hemoglobin, and someone with sickle cell anemia has at position number 6 down there has a different amino acid. So what happens? This is made at the ribosome, and the ribosome folds. Am I not sharing my screen properly? Somebody has a question. Somebody has a friend with sickle cell. Yeah. Depending on where they're from, it's relatively common. It's I don't think it's the most common inherited genetic disease, but it is if you are from sub-Saharan Africa. So you can see normal hemoglobin. We have the proteins made at the ribosome. It folds, makes these proteins. In fact, it has a quaternary structure. It's got 4 protein molecules, and it transports blood. And so there it is. It's carrying the oxygen, and you've got red blood cells that look something like that. Someone with sickle cell. So what happens is that one amino acid is enough to kind of just affect the structure. It's actually hydrophobic, and being hydrophobic, it makes it kind of sticky. So your hemoglobin looks just a little bit different, and you can see they've put a little dent in there to show that it's kind of like a little puzzle piece, I guess you could think of it as. And so what happens is that the hemoglobin molecules will actually form these fibers, and these fibers can't bind oxygen as tightly, and they also change the shape of the cell. And so a sickle, by the way, maybe you've seen pictures of a Grim Reaper, right? And a Grim Reaper has, this big blade that looks something like that, and that's a sickle. It has a couple other names, but that's where the term comes from. So this is just to reiterate that the amino acid sequence is important, and it can affect things biologically, and sometimes in a very significant way. So much more on sickle cell way at the sort of the back end of the course when we start talking about DNA and proteins and things like that. So today we're talking about nucleic acids, and this is the 4th macromolecule, and nucleic acids are made out of nucleotides. So what is a nucleotide? Well, a nucleotide is kind of a complex monomer because it has a sugar. So it has a sugar right there. And that's not a very good picture of a sugar because it should have a bunch of OHs on there, and they're not showing everything on that diagram. It has a phosphate. So we've seen a phosphate before when we talked about the phospholipids. It's the same phosphate. It has a P of phosphorus in it, and then it has a bunch of oxygens around it. And then it has these other things here that are called nitrogenous bases. So nitrogenous bases have lots of nitrogen in them. I think I have some pictures here. There's the nitrogenous bases. So like I said, nitrogenous means nitrogen. So you probably know the codes are the letters of the genetic code. So the C, T, G, and A. And of course, if you are RNA, you're going to have U for uracil instead of T for thymine. So this is just a very brief overview of nucleic acids, by the way. And we'll talk a lot more about them in topic 13 and after that. So you may also know there's DNA or RNA. The R in RNA stands for ribose. So this is the ribose sugar, and you can see it has all those OHs in there. And the D for deoxy in DNA is looking right here. So that is where it is missing an oxygen. So it is a deoxyribose. And so the D for DNA. So a little bit of a difference. Like I said, difference. Much more on this when we get to topic 13. So you probably know what DNA does or have an idea what DNA does. DNA is instructions and it codes for genes, and the genes tell you how to make whatever organism you are. If you're a plant, you're going to have genes for chlorophyll and plant parts, cellulose, genes for cellulose, and making enzymes, and so on. If you're an animal, you're going to have genes for making animal enzymes, animal parts. So DNA is a double-stranded nucleotide, polynucleotide. There's also, sometimes it's called the double helix because it is forms kind of a helical structure. Do not get that mixed up with an alpha helix that we saw in the protein structure. There's also RNA, which is single-stranded, and one other nucleotide that people seem to often forget is not an information nucleotide. Another nucleotide is ATP. So take a look at this. It has all the parts. It's got a nitrogenous base, it's got the sugar, and it's got some phosphate groups. So ATP, of course, is not involved in information like RNA and DNA. It's involved in energy. So I'm just reminded that I meant to go back and take a look at our notes. So we started making this table here. And so we filled it out for lipids and carbohydrates and proteins. So now we should put some information here about nucleic acids. So scroll down here, nucleic acids. The main subunits are nucleotides. And nucleotides, as we mentioned, did I spell that wrong? The spell checker doesn't like it. Oh, my spell checker is spell checking in French. That's always fun. So the nucleotides, they are made out of pentose sugars. They are made out of phosphate groups, and nitrogenous bases. Okay, and sometimes you might find different variations on that for nucleotides, by the way. Sometimes you may find there's no phosphate group, or you might find an unusual nitrogenous base or something like that, but usually they have those three components. This is saying the chemistry. So the chemistry is, what you're looking for, when you're trying to recognize these things. So when you're looking for these things, look for the nitrogenous bases. Maybe I should have put this, this group of stuff here, over here. So it tells you what to look for. And if you're looking for DNA, usually it's kind of a double helix spirally structure. The bonds that connect nucleotides. I didn't mention those yet, but they're called phosphodiester bonds. And I don't expect you to remember that word for Biology 107, but again, if you're interested in chemistry and whatnot, you'll see that term come up. So what is going on with these things? What are their functions? So DNA and RNA, so these carry information. And we have ATP, and we're also going to talk about something called GTP, and these are energy currency, I'll call it. So we'll talk more about ATP and GTP much later on. So like I said, this is mostly an introduction to these things, and kind of at this moment, I want you to kind of know what the differences are on a basic level, mostly that you can recognize these things and use the language of them. So when I'm talking about amino acids, I want you to know we are talking about proteins. When I'm talking about a monosaccharide, you should know that it's a carbohydrate and so on. So hopefully everyone got all the information out of that table. Like I said, I don't usually post this, but if you do want a copy and you missed something, you can always send me an email and I will send it to you, not a problem. So I'm going to go back to the PowerPoint and talk about a few other things, but you may want to load up your Kahoot app because that's what we're going to do in a couple minutes. So one thing that's kind of going to come back again and again in this course is this diagram here. Particularly in the last third of the course, we're going to talk about DNA and RNA and proteins and a whole bunch of different detail. So you're going to see this diagram again and again. And when we talk about viruses, you're going to see this diagram. And so I'm just throwing this up here to, because it's going to come back, and hopefully through repetition, you'll be able to remember some of these things. So just some quick definitions. Replication, that is taking DNA and making a copy of it. Transcription, that is taking DNA and using the genetic code in DNA to make some RNA. So it's very similar to DNA, except for it's shorter, and we're changing all the T's to U's. And translation is where we're taking the code in the RNA, we're taking the triplet codons, and we're converting that to amino acids, and we're doing that at the ribosome. So like I said, you don't have all that right now. Don't worry about it, but when we get to topic 13, after midterm number 2, that's when it's going to get real. So going back to our 2 cell types, right? We're talking about prokaryotic cells and eukaryotic cells. So sometimes I'll say bacterial cells instead of prokaryotic cells, because that's usually what we're talking about. Prokaryotic cells have no compartmentalization. So everything is done in the cytoplasm. So all the DNA and RNA and protein stuff, it all happens basically in the cytoplasm. In the eukaryotic cell, things are a little bit more compartmentalized. We have a nucleus, we have other organelles, and so this is something we're going to talk about as we go over the next few minutes about where things happen and what is going on there in the cell. So one other note or two is that sometimes you'll see these terms combined. So sometimes we're talking about, for example, a lipoprotein. So what is a lipoprotein? It's a lipid and it's a protein. And so, you know, you can see that this is the definition here. So usually the root word, so protein here, that's the bigger part. And then the part that's up front, the lipo part, that's the smaller part. So what you're looking at here is probably a big globular protein like this and a little lipid sticking off the top of it. There's also glycoproteins. And so same idea, you've got a big protein, and then maybe there's a little carbohydrate. There's my little carbohydrate. There's probably a few saccharides on there. Those are supposed to be hexagons, but obviously today I cannot draw a hexagon. So that's called a glycoprotein. The term that means to put a carbohydrate on something is called glycosylation. So sometimes we might say that the protein has been glycosylated. So remember, anytime you see glyco, that means carbohydrate. So what about this last one, proteoglycan? So this is the opposite. The carbohydrate part is bigger. So these are these massive carbohydrates. So here's my massive carbohydrate. I'm just going to make it like a zigzag. And then there's these little peptides that kind of come off the side. So I know this is not an amazing diagram, but the whole idea here is to tell you what a proteoglycan is. And it's a mixture of a carbohydrate with some proteins hanging off of it. So here's our little cell. We've added our DNA to it. We've added proteins to it and a little bit of RNA. So the DNA and RNA are those squigglies in there. And that's going to help it to remember and help it to give information to its daughter cells. So there's our cell there. And kind of going back to the beginning of the unit where I talked about all of the parts of the cell. Okay, so I have a Kahoot for you. And so hopefully this one's going to be a little bit hard because, you know, unless you're really familiar with the chemistry, but we'll go through this and hopefully you have a little bit of fun as you're quizzing yourself. So load up the app and I will load up the game. So here we go. It's loading. Oh, I have to hit start. Sorry about that. So there we go. I'll give you 20 seconds to join. Okay, here we go. Question 1, what category of macromolecule does this belong to? Only 10 seconds, got to answer quickly. So that was a lipid. Let's just take a quick look at the diagram. That, by the way, is, that's not cholesterol, I think that's testosterone. And so that is a steroid, and the steroids are lipids because they're mostly carbon and hydrogen. 2 oxygens only and a whole bunch of carbons in there. All right, so question 2, what category of macromolecule does this belong to? So what is in there? Use your chart, take a look at what atoms you have and what functional groups. All right, I warned you this one was going to be tough. This is an amino acid. So if you take a look at that, on the left we have the N for nitrogen, NH2, that's an amine group. On the right of that I have a C and 2 oxygens and a hydrogen, that's a carboxylic acid group. So this is an amino acid. So like I said, I told you this one would be hard, and maybe we'll do another quiz next week if we have time. But something you will need to do is study these for the test. Okay, question 3. What category of macromolecule does this belong to? Maybe this one's a little easier. We just saw it a few minutes ago. Okay, well done. 6 people got the right answer. This is a nucleic acid. Well, this is actually just a nitrogenous base. And I did not color all the nitrogens to make it obvious. That actually is how it was colored when I found the diagram. But this one here is adenine. Okay, question 4. Here's another one. Long, skinny one with lots of carbon and lots of hydrogen. Maybe that's a hint. There we go. A few more people getting this one. This is a lipid. Lots of mostly hydrogen and carbon. I see some people got the wrong answer saying it was a carbohydrate. Carbohydrates would have many more OHs, many more alcohol groups on that. Usually the rule for carbohydrates is there's usually almost as many OHs as there are carbons, usually 1 or 2 less. So in this case here, there's like, what, 18 carbons? So there should be at least 16 OHs on there. So it's not a carbohydrate. Okay, a few more questions if you want to redeem yourself. Okay, so here's another one. Sorry, the diagram's a little small, but maybe you can see the letters in there. Hopefully that is a big clue. So that's a nucleic acid. So notice all the letters in there. We've got A's and U's and C's and G's. A few other funny things in there, but this is transfer RNA. So this is a nucleic acid. Okay, so the scoreboard is getting mixed up a little bit. That's good. 2 more, 3 more. Okay, how about this one here? I know I showed you this one last day. 9 out of 9, carbohydrate, excellent. That here is sucrose. This is table sugar. So that is glucose on the left and fructose on the right, and you combine them together and you get sucrose. Okay, 2 more. So what about this thing? It's got a lot of swirly, cartoony things in it. We definitely talked about this last day. Okay, so half of you said protein, which was correct, and half of you said nucleic acid. So this is a protein. Whenever you see these ribbon structures, this is representing an alpha helix. So a DNA double helix would be exactly that. It would be double. You'd see 2 strands wrapped around each other. So I'm not sure if that's where the confusion was. Okay, I think there's one more question. A few people rooting for the top spot. So what is that? Okay, most you got it correct, and that is a nucleic acid. This here is, you can see the phosphate here. You've got a carbohydrate here, and you've got a nitrogenous base, so that would make it a nucleic acid. So let's see how the scoreboard came out. Bronze we have with 4,000 points, Levi. Silver for Tristan, well done. And who's it going to be? Mina, well done. 4,345 points. And runner-up, congratulations to Matt and Trevor. So I warned you that was going to be difficult. That some of these structures, unless you're looking at them all the time like I do, and you're really familiar with them, it takes a little bit of practice to learn what they are. So I was hoping topic 2 would be kind of combination of review and maybe some new stuff at the same time. So anyway, we're going to move on, and I'm just trying to get my screen to share here. And as I mentioned before, we're going to move on, and we are going to actually skip right to topic 20. I'm doing them slightly out of sequence this semester. I wanted to talk about viruses sooner this semester. It's usually the very last unit. And it certainly does help to understand a bit more about DNA to understand viruses. But I do believe that talking about viruses this year in particular is very important. And I want to talk about vaccines and a few other things. So I'm hoping you will learn something. And I am hoping that as somebody taking a science class, that you will go and tell people about it as well. Someone's asking if this PowerPoint is on Moodle. I don't know. Sorry, Emma, if I forgot to put it up there. I've been trying to get them up there ahead of time, but I think maybe I forgot to put it up. So I will get it up there after class. But you do have the notes for this topic, and it will cover the same material. So like I said, I'm hoping that you will take some of this information home with you and tell people about it. Unfortunately, I see there's a lot of misinformation about viruses out there, a lot of misinformation about vaccines and those kind of things. And I think it is our job as people who are learning about this stuff to tell people about it, you know, what viruses are and how they're affecting us because of all that misinformation out there. So let's get into this. So usually I ask people, have you ever had a viral infection? And most people, you know, kind of hum and haw, and you know, we're pretty much all sure we've had one, whether you know it or not. And then I ask people this question, and I say, has anyone ever had a herpes infection? And of course, I say very quickly, you don't need to put your hand up on that one. But I'm here to actually tell you that you have had a herpes infection, and let me explain what I'm talking about. So if you take a look at this, this is a chart that talks about all the herpes and viruses that can infect humans. So there's 8 that are called human herpes viruses, 1 through 8, and there's something called a B virus as well. And the ones that people are probably most familiar with are these 2 here. So they're called herpes simplex 1. Right there, and herpes simplex 2. So herpes simplex 1 usually is the culprit that causes cold sores. Sometimes herpes simplex 1 will also cause genital herpes. Herpes simplex 2 is kind of the opposite, where it's usually the culprit that causes genital herpes and sometimes can cause cold sores. So you've probably heard of cold sores, you've probably heard of genital herpes, and they can be caused by these 2 viruses. And usually when people say herpes, they do mean genital herpes, the sexually transmitted infection. But medically, when we say herpes, it could actually mean quite a few different viruses, or at least when we say herpes virus infection. So I want to point you also to the numbers on the right. And there's been a number of studies that look at how many people have had these things, because one thing about herpes virus is that they are lifelong infections. So that means that when you get infected by a herpes virus, it is with you for the rest of your life. So if we were to look at our class, and there's about 10 of us, I think, so about 6 of us probably have infections by herpes simplex 1. So the prevalence is 60%. Herpes simplex 2, the prevalence is about 10 to 20%, depending on where you are in the world and how old you are and all those kind of things. So at the moment, there's a good chance that most of us, at least the majority, at least more than half of us, have at least one of these viruses. What about herpes, human herpes virus 3 and 4? These actually have their own fancy names. There's varicella virus and Epstein-Barr virus, so they're kind of fancy medical names that you don't necessarily know unless you know a lot about viruses like me. These cause things like chickenpox and infectious mononucleosis. So this is something interesting here, actually, this virus that causes chickenpox, because it used to be a rite of passage. 95% of us would get it by the time we were 10 years old. Nowadays, you guys most of you are young enough that you probably had the vaccine for it, and probably will never get chickenpox. Infectious mononucleosis, same thing. It's a rite of passage. Usually by the time you're 30, you've had a 95% chance of getting it. So I see somebody has a question here about is it passed on through birth? They're talking about, I think, herpes simplex viruses. So herpes simplex 1 is usually passed on after birth and just by close contact with people. You can imagine somebody might have it and kiss you as a baby or something like that. So usually you get it as a child. Herpes simplex 2, if you're getting genital herpes, it's usually when people become sexually active is when they're likely to get it. Infectious mononucleosis, by the way, is also spread by mucus and respiratory droplets, and sometimes it's called the kissing disease because sometimes people will get it through exchanging saliva. So what about these other ones here? There's a whole bunch of herpes viruses out there. These ones you may or may not have heard of. They're very common to get and very common that you don't have symptoms. All of these here are kind of the things that might be risky if you are immunocompromised, or maybe you're getting organ transplants or something like that. This one here, sixth disease, is sometimes called baby measles, and sometimes babies will end up with a little bit of fever from it, and sometimes even a little bit of a rash, but it's usually one of those things that, you keep your baby at home for a day and don't send it to daycare, and no one thinks anything of it because it's so mild. So looking at this, you probably had a 100% chance of actually getting herpes human virus number 6. So there's a very good chance that most of us here are infected by probably a minimum of 3 different herpes viruses. So that's something you can tell your family today and be proud of it, because it doesn't mean anything. It just means you're human. It doesn't mean that you're sick or anything like that. It just means that you've been in the human population long enough to have been exposed to these things. Now, the last virus on the list, the B virus, I'm assuming none of you have had herpes B virus. This is a monkey virus. It is 100% fatal, and if you have it now, you will probably die, and I hope that's not the case. Anyway, so what is this all about? What I'm trying to do is tell you that viruses are common, and they're everywhere, and sometimes they're doing bad things to us, and many times they're not. They're the things that are barely an inconvenience. Here's some virus facts for you. So viruses infect every living thing, at least that we know of. Pretty much everywhere we look, every organism we've ever looked at, it seems that there's at least one virus that infects it. This seems to be the case anyway, and we find them everywhere. You just have to look for them. We are exposed to billions of them daily. We breathe air, we touch doorknobs. Pretty much everywhere we go, there are viruses, and we are being exposed to them. I'm showing you these vegetables because maybe you had a salad for lunch today, and on that salad, there would have been viruses there that would have been left by the insects that walked on those vegetables when they were out on a farm somewhere. And they're not doing anything to you, right? They're not making you sick. The organisms that live in us, we have E. coli in our intestine, and E. coli has viruses that infect E. coli. And so viruses are everywhere. And kind of the bottom line is here that the majority of viruses we are exposed to have little or no impact on our health. They don't infect humans, or our immune system takes care of them, and they're not necessarily something to be scared of. They're a normal part of the biosphere. And I think viruses are very cool and exciting, and I really enjoy learning about them. And of course, some are to be feared, and some we need to take. We need to do things medically with them, vaccines or prevention. And I don't wish bad infections upon anybody, but I do think they're very interesting because they're very small and simple things. And it kind of leads into our next point is what is a virus? So there's a definition I pulled out of, I don't think it was our textbook, I pulled out of a different textbook, but you can see it's covering a few things there. It says it's small, minuscule means small. It's acellular, it doesn't have a cell. And basically it's some nucleic acids surrounded by a protein coat. So that's it. We're talking about a little bit of RNA or a little bit of DNA, and it's packaged in a protein coat. And that's it. And so what are they doing? They're tricking cells. They're going into cells and they're bringing those genetic instructions, and the genetic instructions say make more virus. And that's really what they do. And we're going to talk about that a little bit, just some details in terms of how exactly viruses do just that. And you can see there's a little picture there showing a virus infecting a cell, and it's bringing its DNA in there, and it's making new viruses using those instructions in the DNA. So let's just take a look at this word minuscule for a minute, because I think it's really fascinating to think how small these things are. Most of us can't think that small, right? So here's a human. This is actually a big human, 2 meters. That's taller than 6 feet tall. 6 feet tall is probably about 100 and I'm thinking 85 meters. So this is a tall person, but just to make the math easy. And let's take a look at a rhinovirus, which is 200 nanometers. So a rhinovirus. So rhino is like this. So this is the point where usually in class I ask people, what does a rhino mean? And rhino people are thinking about the mammal, and it has a big nose, and it has a horn on its nose, and in fact rhino means nose. So this is a virus that infects your nose. This is a virus, and it's the most common cause of the common cold. So there's what a rhinovirus is. So if we do some math, we can see how big this is. 2 meters divided by 200 nanometers. So I got to convert, of course, meters to nanometers. So a nanometer is 1 billionth of a meter. So I had to multiply 2 times a billion. So 2 billion divided by 200 nanometers, that's about 10 million times. So I know a million. That is, that number is too big for most of us to count to in a reasonable amount of time. It's very hard to conceive. So I saw this thing on Cells Alive that talked about comparing a virus to the head of a pin. So you can probably imagine a pin, and that's about 2 millimeters in diameter. And a millimeter is 1,000 micrometers, or a micron, as you can see on there. And let's compare that to a couple of things that we can see. So a dust mite is about 300 micrometers and just barely visible by the eye. And a strand of hair, same thing, is just barely visible by your eye. Anything smaller than the thickness of a strand of hair, we can't see it. So compare that to a red blood cell, which is about 10 micrometers. So that's 1/10 of the size of a strand of hair. It's too small for us to see with our eyeball. We need a microscope for that. So how big is a virus compared to a red blood cell? So it's not going to show up here. Okay, if a red blood cell looks like a speck, the rhinovirus is going to be a very small part of that speck. So I zoomed in, and if you look at a red blood cell in terms of that size, the virus itself is like a speck compared to that red blood cell. This can only be seen by an electron microscope. We can't see this with a normal microscope viruses, by the way. So how big is that compared to the pin? About 500 million of these viruses would fit on that pin. That's a big number. If you think about it, one sneeze when you have the cold could infect easily thousands of people. So that's how small a virus is, very, very tiny. And it's amazing how something so small can affect us so greatly. So here's another way we can compare the size of these things, right? So we can compare, let's say, human cell, so eukaryotic cell. So human cell is eukaryote, to bacterial cell, which is a prokaryote. And then a virus, of course, is acellular, so has no cell. So the human cell, depending on which cell type it is, a red blood cell versus a neuron or whatever, is maybe 50 or 100 micrometers. Genome size, this is our haploid genome. We've got about 3 billion base pairs and somewhere around 20,000 genes. If you take a look at something like E. coli in terms of size is about 1/100th of the size of a human cell. They have about 4 million base pairs. Okay, so that's an entire order of magnitude, 1,000 times smaller in terms of the genome than a human cell, and somewhere around 4,000 to 5,000 genes. A virus, way smaller than that. So as I mentioned, the rhinovirus is pretty big. It's 200 nanometers. Many viruses are actually much smaller. And we're looking at anywhere from, a dozen to 24 genes is pretty typical for most viruses. So very, very, very small things. So there's this picture. I think I showed you this one in topic 1, showing the eukaryotic cell on the left and the prokaryotic cell on the right. And I have the influenza virus shown there as a tiny, tiny speck. And we zoom in on that, and there's an electron micrograph of that influenza virus. And you can see that it doesn't have all the organelles. This virus does have sort of like a membrane. I don't like to use that word membrane. There's a better word for it, and I'll explain that in a moment. And it has DNA or RNA. So one other thing to compare, I just want to compare a few other features that we've discussed a little bit between animal cells, bacterial cells, and viruses in terms of what they are. So you can see the first note there is an intracellular parasite, meaning does it live in a cell? So maybe I'll write that. Does it live inside a cell? So animal cells, maybe I should have put in eukaryotic cells. No, not usually. There's a few parasites that might have parts of their life cycle where they live intracellularly, and you can see the example there I have is the malaria parasite. But usually an animal cell, you feed it food and it eats and it grows, right? Same thing with a bacterial cell. Most of them are free living. They live, out in the environment and they ingest nutrients and they grow. There are some exceptions. So chlamydia, that's a sexually transmitted disease, is a bacterial cell that actually does live intracellularly, but it's kind of an exception. It's actually, believe it or not, we used to think it was a virus until the modern genomic era, where we could actually take a better look at it. Viruses, on the other hand, yes, they do live intracellularly. They do have to go inside a cell in order to replicate. You can see the next one says, does it have a plasma membrane? Animal cells, yes, they are cells. Bacterial cells, yes, they are cells. Viruses, no, they don't have true membranes. And I'll talk about what they might have in a minute. Do they have both RNA and DNA? Animal cells, yes. Bacterial cells, yes. Viruses, no, they have DNA or RNA, not both. And we'll talk about that in a moment. Ribosomes, that's a yes, yes, no as well. Viruses do not have ribosomes. They do not make their own proteins. They cannot do that. Making ATP, again, yes, yes, no. Animal cells and bacterial cells, they consume food and make energy, and viruses do not. Last one, sensitive to antibiotics. Okay, that one's different. This is a no, yes, no. We're going to talk about antibiotics in topic, is it 4 or 5? I can't remember which one is on bacteria. And antibiotics are something usually we take to treat bacterial infections, not viral infections. So more on antibiotics later in topic 5. So let's talk a little bit about virus structure, and we'll see how far we get today. We're not going to get a chance to talk about COVID-19 today, but just a little bit more general stuff about viruses. So I mentioned about viruses that they are basically some genetic instructions wrapped with a protein coat. So there's our genetic instructions. We can have DNA or RNA. And the interesting thing about viruses is that the DNA could be double-stranded or single-stranded. The RNA also could be double-stranded or single-stranded, which is kind of weird because we don't see that kind of variety in what's going on with nucleic acids otherwise. So there's our protein coat. And our protein coat has a special name, and it is called the capsid. So sometimes people will use, and they'll say nucleocapsid. But what does it matter? It's still a capsid. By nucleo, it means it's surrounding the nucleotides. I don't know why some people would use a longer word when you can use a simpler word. But hey, you know, that's just the way people are. Sometimes they like to be complicated. So that's it. That's actually what a lot of viruses are. They're just bad news inside a protein coat. But some are a little bit more complex. Some of them have kind of like a membrane. We actually used a different word. We used the word envelope. And this is not found in all viruses. This is what they might take on their way out. So when they're leaving their host cell, they might grab a little bit of, they might grab a phospholipid bilayer. It's not technically a membrane because it's not doing normal functions of a membrane. It's not like transporting materials or anything like that. Usually a part of that membrane are some virus glycoproteins. And sometimes in the case of this coronavirus, we're also calling these spikes. And sometimes they kind of look spiky, so sometimes they just call them spike proteins. But really they're viral glycoproteins is what they are. So that's kind of your basic virus structure. I'll show you some pictures of a few different ones here now and show you, you know, all the variety that you can find in viral structures. So here's a few different ones, and if you look at those things we just talked about, there's the genomes, so the DNA or RNA. They all have capsids surrounding that, and the one on the right has the envelope as well. So I'll show you some, like I said, some different viruses here and different examples. So some viruses we call helical viruses, and you can see really what that is that the genome is wrapped in sort of a tubular or a cylindrical type of structure of capsid proteins. So two examples there is tobacco mosaic virus. And the big one that we were talking about a lot a few years ago is, of course, the Ebola virus. And you can see it's long and skinny enough that it will actually tangle itself and form a knot. And I'm not sure whether that has any actual biological function or if it's just because it's sloppy. Some viruses are polyhedral. A term that's often used by virologists is this term here, icosahedron, and that means that you kind of have a bunch of triangles. So the capsid proteins form these triangles, and the triangles kind of snap together in a, you know, not necessarily round, but a polyhedral shape. And so often you'll see viruses represented as these polyhedral shapes. And you can see this one has a few little spike proteins growing off the side. So this is actually a really big class of viruses here that look just like this. So you can see some examples of adenoviruses. Some of those cause the cold as well, and poliovirus, which causes polio. A bunch of animal viruses are enveloped, and you can see this one here on the inside. The inside of this one here, this is actually got the helical shape on the inside, and then it's surrounded by that that envelope and those glycoproteins. So I have three examples there. I have the HIV, which is the virus that causes AIDS, so human immunodeficiency virus. I have the influenza virus, and I have the SARS coronavirus 2, which is the one giving us all the trouble this year. So in my notes, I have information both on HIV and influenza. I'm probably not going to be covering them next day, but they are in your notes if you're interested in them. They're very important viruses to human health. But this year, the focus is, of course, on SARS-CoV-2, which is SARS coronavirus. So there are a few viruses out there that are even more fancy and complex, meaning they have basically extra features. And you know, there's a few out there. This is one of the more interesting ones. This is something called a bacteriophage. So bacteriophage, by the way, that means bacteria, and phage means eating or eater. So this really just means eater of bacteria. And so this is a virus that will infect E. coli and other bacteria. So you can see that the, if you just take a look at this part here, the head is just a polyhedral virus. It has DNA, and there's the protein coat, the capsid, but it has this big tail structure on it, which is actually a helical structure, and then these tail fibers. So it kind of looks a little bit like some sort of, you know, you can imagine this is like a Mars lander, and it's landing on the planet and crawling around. But it's actually a little bit more like a big syringe. And so this syringe is going to dock onto E. coli down here. and it's going to basically inject its nucleotides right into E. coli once it burrows through the E. coli cell wall. So that's a bacteriophage. And of course, there's the internet. So I was looking for a nice picture of a bacteriophage, and what did I find? I found this guy here. So the internet, you know, always comes through with lots of weird things. You can depend on it for that. So viruses come in all shapes and sizes, and you can see there's a few there. Maybe you recognize a few of these. There's the influenza virus and HIV. You can see rabies looks a little bit, it's kind of like a bullet-shaped virus. Smallpox virus is that big one on the left, and here's the bacteriophage infecting E. coli. You can get an idea of the idea of size of these. They do come in many, many shapes and sizes. You can see the smallest one there is a flavivirus. So that would include the one that causes yellow fever, but also like West Nile virus. And it's very, very tiny, almost as small as, you know, only a few times larger than hemoglobin. So I just want to finish off today kind of talking about the abundance of viruses out there, because this is something that has fascinated me for years, because as I mentioned, we seem to find them everywhere, and they seem to infect everything. And so they must be having a massive impact on our biosphere. And just to give you some ideas of some numbers. So let's take a look at one virus. Now, one virus here, HIV, is a human immunodeficiency virus, so it affects only humans. So here's the numbers. There's about 34 million people infected with HIV currently. And so what does that mean for how many viruses are out there? So if you look at kind of the average of how many viruses these people have in them and multiply that by 34 million, you get about 10 to the 16. So that's a 1 with 16 zeros behind it. So that's how many virus particles of HIV are on the planet. So that is a massive, massive, massive number. And now think about it. There's other human viruses, and then there's organisms that aren't humans. So we could add in all the whale viruses and all the plant viruses and you name it. The number is staggering, and lots of people have tried to estimate these kind of things. I'll show you one number that was estimated in terms of how many bacteriophage are in the ocean. So marine biologists, particularly marine microbiologists, will take seawater, and you don't see much sometimes unless you put it under an electron microscope. And there's been a few people that have taken a look at some and come up with some numbers. And so take a look, 50 million. What do you think that is? That's the number of viruses per milliliter of seawater. And there's been a whole bunch of studies on this. And sometimes people come with a bit of a smaller number, like 20 million. There's been a few people that have much larger numbers. But this is the number that seems to hover around. So a milliliter of seawater, that's about the amount that, about the size of a sugar cube, right? So multiply that by the amount of water in the ocean. We're getting 10 to the 30. This number is like, it's so big, your brain cannot comprehend it. I'll show you how big this number is, because I saw this, there's somebody talking at a conference about this, and we're talking about estimating this kind of number. So that's about 10 to the 23 infections per second, which is about Avogadro's number, if you remember your chemistry, which is a massive, massive number. But how big is 10 to the 30? So let's go back to our little virus here, and let's assume it's about 200 nanometers. That was the number that I gave you before of the rhinovirus, but it's pretty true for a bacteriophage. So what if I took 10 to the 30 bacteriophage and I just kind of stack them and put them all together, and how far would that reach? So you can do the math, right? You can multiply 10 to the 30 times 200 nanometers, and you get 200 million light years. I know this is like, I cannot conceive this. This is huge. I checked the math myself. You can check out this website that talks about this. By the way, the closest galaxy to ours is Andromeda, and it's only 2.5 million light years away. So this is a massive, massive number. And that's how many viruses out there. And like I said, they must be doing something out there in the ecosystem. And I think it's a very, very fascinating thing to think about. So I'm just going to finish off today about going back to Unit 1 where we talked about our viruses alive. I think I showed you this exact slide before, and we're talking about cells, and cells, they carry out all the properties of life. They're organized, they reproduce, they grow, they use energy, they respond to the environment, they adapt, and so on. Viruses, on the other hand, they are organized, yes. They reproduce, well, they require a host, so that's kind of a yes, no kind of answer. Do they grow? Not, well, sort of a yes, no answer. Do they use energy? No. Respond to the environment? No. Homeostasis? No. Do they adapt? Yes. They do mutate, right? So are viruses alive? Usually we would say no, because they don't fulfill all the requirements of life. Some people would say they're alive once they're in a cell, so it's kind of a gray zone. They're a very important part of the ecosystem. And biology, but they're not made out of cells, they infect cells. So we are done for today. We're going to come back to this unit next day, and we're definitely going to start talking a little bit about SARS-CoV-2 and COVID-19. And I'm not sure if we're going to quite get to it next day, but I'm going to finish off on that, talking about how you make a vaccine, what a vaccine is, and how does it work, and all those kind of things. So we'll leave that for next day. Hopefully everyone has had a great weekend and you're having a good Monday, and I will see you on Wednesday.